So, hello Gurnoor, let me officially welcome you, dear.
Thank you, ma'am.
It's a privilege to hear from our alumni and the person who achieved so much in this short span of time. I'm very happy being a faculty, being a part of your journey when you with us. Now, my major concern is like, dear, there are many freshers who might have seen you for the first time and they might have heard you about first time. So I need your interaction with them. And the concern is that I want you please kindly stick with the presentation for 25 minutes so that they could get enough time to talk to you, because that's what they want more.
Surely.
And in case we get off, then we'll again reconnect if something will left here.
Surely.
Okay, so welcome Gurnoor, respected all faculty members, HOD sir, and my all students who've been a part of this webinar on this Sunday. Gurnoor, it's always been a privilege to have you here with us, and I remember your visits to JUIT. You are one of the brilliant students of JUIT and of Department of Biotechnology and Bioinformatics. Your bachelor's is in Bioinformatics, and which definitely opened many avenues for other aspiring candidates who are in second year, third year, or in the coming fourth year with us. So Gurnoor, please kindly start with your presentation. We would like to hear more from you.
Okay, just want to interrupt here.
Yes,
this
sir.
is like for everybody. I would like to congratulate Gurnoor for getting a very recent and new position in Denmark. So he is going to soon join a very good industry in Denmark. So congratulations, Gurnoor, from all of us.
Congratulations, congratulations, congratulations.
Congratulations.
Thank you, sir. My slides are visible?
Yes.
Yes.
Perfect. Okay. Then I'll just start. Yeah, I'll be talking about FAIR data principles, FAIR genomes, Chrosomics data integration. And yeah, ma'am gave a very nice introduction about myself. Yeah, I did my bachelor's in bioinformatics in Jaypee University. And I did my bachelor thesis then with Tirthraj sir. And it was one of my first milestones, I would say, in life. I did also, I was very happy doing that thesis. We also presented it in Berlin in an international conference. And then I was doing my master's in life science informatics from the University of Bonn, Germany. After that, I chose to do Ph.D. in the Netherlands. And I, during this time, I also work, like I currently work in Nijmegen in the Radboud University Medical Center as a data steward. While my PhD, I was working as a research associate and also worked in a software company, a research-based software company called Fraunhofer Sky as a software developer. And some of my skills are focused towards Java and Python, text mining, DevOps engineer, and data engineering. So currently I work as a research data steward in a medical hospital. And so one of the interesting questions I'm always asked, it's a very new term, what is a research data steward? So if you talk about three different sectors in which a research data steward works, one is towards policy making, one is towards research, and then one is towards facilitating, making an infrastructure, so infrastructure facilitator. And the policymaker basically works with the data guidelines, whether it's FAIR data guidelines, GDPR guidelines, for universities, for research institutes. The researcher actually is like a DevOps engineer who basically deploys data, workflows, tools, standards, and infrastructure. And then the infrastructure facilitator is the one who takes care of software and hardware services and the technical infrastructure. Like if you're using a cloud, how do you publish things on the cloud? So I'm mostly like towards the right side of this circle. I'm half, I would say, towards research and half towards infrastructure development. And one of the big shift in research today is that the importance of data is growing more and more. So we need to not just publish an article, but we want to have a significant proof, a data proof in that article that this is worth what we are trying to claim, and these results are reproducible. So in general, in a research data life cycle, you create a data, which could be like a sequencing pipeline or a raw form of data. And then you make a hypothesis, you process that data, you analyze your hypothesis, and then you preserve your data. At this point, most of us publish our scientific publications, and then but an important part to continue further is giving access to this data, and then making this data reusable. So that completes the complete research data life cycle. And today I'll be also mostly focusing about how do you preserve your data, how do you make it reusable. One of the important terms that came recently in a conference in 2015 was FAIR data principles.
कुछ भी हो जाए वहाँ इधर सोएगा नहीं जी है नहीं उधर पूरा फाइल खाली होता है फिर भी इधर मरना है साले ने वो कोई बांगलू
Okay, FAIR data principles, which are a set of principles which ensure that data are shared in a way that it is reusable for both humans and the machines. FAIR is an acronym which stands for Findable, Accessible, Interoperable, and Reusable. So what does findable actually means? Findable means that both our data and metadata is easily findable, not just by humans, but also by computers. Let me give you an example. If you're measuring regression, if I as a researcher is measuring, has a regression equation, I can't just publish my results by saying this is X, this is Y, and this was my equation. I need to, I cannot even just like define X as my explanatory variable, Y is my dependent variable, but I need to define what actually I'm trying to compute, whether it's, for example, if I'm trying to measure the average weight of a person based on his height, I need to really mention that height and not just H1 or something which recognizes height by both human and the machine. So it should be an identifier to which points out that this is height of an individual, and W stands for weight of an individual, but we should present with an identifier. And machine readable metadata is essential for this automatic discovery of relevant data sets and services. Coming to accessibility, by accessibility, I don't mean that every data should be open. Of course, sensitive data should not be open, but the accessibility of data should always be well defined, which means that license information of the data, the restrictions of the use of data, or the procedure of querying the data, the protocols need to be followed, and the copyrights of this data. Coming to interoperability, interoperability is one of the non-trivial points and one of the most creative efforts in making data fair. And interoperability means that if I give you data in, for example, CSV files, and I want it in JSON or XML, that should be possible. So the data should be defined that way, that if I have CSV files, if I like to plot a chart, if I like to have it as JSON files, so we define really a way that data can be automatically combined with other data sets and also be taken from one format to other. And when we do this, we define the data and metadata sufficiently so that both humans and computers can reuse it and combine it with other data in future research. So these fair data principles got a lot of, I would say, recognition, even in one of the global summits. President Obama also said that, yes, fair data principles is one of the future way of capturing data and saving data for the future. But let's look at what FAIR is not, because that makes us understand better what FAIR is. FAIR is no standard. So it's not any standard in which we are capturing data. It's not a CSV file. It's not an XML file. It's not a JSON. It's not a new standard. It is actually a set of guidelines that ensure that data is reusable. And then FAIR is not equal to open data, and open is not equivalent to free data. So in FAIR, defining the protocol of usage of data, license information is very important. And FAIR principles do not by themselves cover the crucial aspect of intrinsic data quality or ethics. So FAIR doesn't compare what the data is about, the data quality, but it is just the standard to publish data. Traditionally, just to give an example of what FAIR is, traditionally we used to save data as relational databases. And in databases, as most of you, I think so, would have now studied about databases, we have tables and we have columns which define what the data is about, like metadata in columns. And then we have a structure in this tables, and maybe you also guys have studied about normalization of databases. Like there is definitely a limit of every database. And one of the examples I show that, for example, when one of you guys would start, like after, when you'll get your first job, get placed, you would move to a different city, and then you would register in that city. And then you would register in your new bank account for receiving your salary. You would think of having a health insurance. You would think of registering to a doctor. You would have an employer, and then you would also register to income tax department. Most of these things would have one parameter of yours common, which is address, which is actually a parameter which is actually able to find you. But it's a very common thing that many of us have kind of two addresses, whether it's one is a permanent address, one is a local address. Also in our housings in JUIT, like in our hostels, we have rooms which is kind of our address at a time where we are living, but then we also have a permanent address. So how do we, when we capture this information in databases, it's a single component which is not really able to capture this properly. And this is like a problem. Sometimes like there are people in Europe which live on a tiny house, which is a house on wheels. How do you capture that information? So one of the conceptual things that came out was Semantic Web, to capture this information, that we don't publish data in databases. Now we think of publishing data in data graphs. And in a graph, we mostly have a subject, a predicate, and an object. There are three components of a graph. Here, like the subject is the person for whom we are talking about. Object is the house, and then predicate is basically the relationship. And this concept of semantic web actually came from the person who created internet, Tim Berners-Lee. He said that instead of just capturing, making a web of web pages, an internet of web pages, we should now start making internet of data. And here, so basically when we define a subject, predicate, or an object, we basically define that with the help of URIs, which is like uniform resource identifiers. These are just like URLs to find web pages, but here this annotates the identifier of a person. And so Tim Berners-Lee was the person who made internet. and also who proposed the idea of linked data or linked open data. And he basically made 5 stars to measure how your data is linked, how linked open your data is. So the first star is basically that you have data on the web. The second star is that your data is machine readable and is in a non-proprietary format. And then you have RDF standards, which I just showed, like Semantic Web. This is the standard is called RDF. And then once you have a linked RDF, that's a 5-star data. And if you see, like, if ever you wonder, like, you know, how my contacts from WhatsApp come in my Gmail, or my contacts from my Gmail give me a notification in Facebook that, oh, people you may know, that's what the logical thing is happening. We are standardizing data and semantifying data in a way that we can actually communicate between different data resources. And one of the ways to standardize this data is using ontologies. Many of you might be aware of the gene ontology, which is an ontology to capture biological processes, molecular functions, or cellular locations in regards to a gene. And then there is like sequence ontology. So basically ontology is just a formal representation, a machine readable structural representation of a concept. And in like biological processes, it's many times hierarchical based. So we have like behavior based biological processes, cell development based processes, cellular processes. So we are capturing data in a formal representation, which is machine readable. Many, I have different examples of disease and other phenotype ontology, like the disease ontology, HPO, the human phenotype ontology, which is really used, mammalian phenotype ontology, experimental factor ontology, which is coming from EBI, and they capture all the things related to a experiment. For clinical terms, we have like LONIC, ICD-10, SNOMED-CT, and different NCIT for capturing cancer terms. And what does interoperability basically does, I would like to show you in a form of analogy, which is like whenever we travel abroad, we always carry a travel adapter. So what it does is basically if you have, if you're living in Europe and you're traveling to India, you need a travel adapter to connect your electronic appliances. So basically it's doing, so if you're capturing data in KB terms and you want the pipeline in which you want to do an analysis is doing it in KEGG terms. So basically you use ontological methods to capture data, and then you can map data from one format to other. One of the examples from my project I show here, which is like I annotate an individual. So how do I capture data in the form of an RDF graph and schematic overview, and then also a validator. So basically if an individual is annotated by an identifier and an alternative identifier, with the help of RDF, I define that this individual is a type of Homo sapien, which denotes an identifier, which is a type of an identifier, and then this is the value of an identifier. And similarly, the same person has an alternative ID, which is of the type alternative ID, and this is the patient ID. So this, while I model in schematic overview, I also formulate these rules in a form of a RDF validator, which is called check, and then I publish the data, which is the RDF on the top right. So what is FAIR data in general? We capture data, metadata with the help of standardization, ontologies, and uniform resource identifier. We make machine readable data. We release this data mostly with APIs, which is like a single line code to query anything. API stands for Application Programming Interface. And then we have the FAIR access point. So many big initiatives worldwide are understanding the value of FAIR data. One of them is GoFAIR, which is the international initiative. One is Elixir, which is a European bioinformatics initiative that we have to publish all the European bioinformatic resources in FAIR. Then we have the Personal Health Train. You can also see videos about these different initiatives on YouTube as well. And Personal Health Train is a Dutch initiative where we want to basically optimize the whole workflow of a person going to the doctor, getting the analysis, getting the results done, and getting the report back, and in a fair way. Then we have for research, we have the Chrosomics, Netherlands X-omics initiative, where we are capturing different omics like metabolomics, genomics, proteomics, and trying to capture that data in FAIR with the European Joint Program for Rare Diseases and the European Infrastructure for Translation Medicine. And one of the big initiatives globally, like the GA for GH, Global Alliance for Genomic Health, and 1 million genomics as well, which is a big European project. And but then we had a certain change in our lives when we were exposed to the pandemic, the coronavirus pandemic. And it's also a learning for us, I would say, at the time of crisis, that we should not let this crisis go waste. So all the big research grants from Netherlands, from Europe, and most big research grants started making a data management plan where their core method is that we want data to be fair as much as possible. So now the focus, instead of having a publication, we want to publish data, basically. Coming to the next topic, I hope I'm not too slow. Coming to the next topic, which is the FAIRgenomes. I'll just pick up speed now, which was one of the projects where I'm a lead developer. And here we are trying to make national guidelines to promote reuse of NGS data in research and healthcare. And this project, basically, we have about partners from 14 different institutes and all the medical hospitals in Netherlands, and infrastructure-based hospitals, bioinformatic-based research centers. And FAIRgenomes is basically that from patient information to sample information to materials information to clinical information to reporting, analysis information, sample preparation information. We want to capture all what we are doing and define these rules so that we can automate this process. And before having FAIRgenomes, we have individual databases. We can answer like simple questions like what was the phenotype of the patient, what kind of sharing is allowed by the consent, which tissues were sampled, which sample preparation kit was used, and so on. But if we have FAIR, we can do a internet query through all the hospitals and see that give me all the VCF files for a certain disease regarding this research study, and which were done through the HiSec machine processed with GATK4. So this would be possible. And yeah, again, we are 61 people from 14 institutes, and we are trying to define metadata, which is captured in the whole pipeline. And we are interacting again with different partners so that we can use the FAIRgenomes project to capture genomics data. And it is a very modular based structure. We capture study information, personal information, consent information, whether it's a general consent or an individual consent, clinical information, materials information, the sample preparation and sequencing and analysis. And the way we release this data is with code books, which are like data catalogs. So an example of personal information code book. In personal information, we are capturing an individual ID, gender, genotypic sex, karyotypic sex, country of birth, ethnicity, and so on. And if you see, like, so individual ID, according to like the GDPR guidelines, patient ID is a very sensitive information. In Europe, we cannot really exchange this, especially like when there is a traceability of the person. So this is like, these are just rules right now to define how shall we capture data and how shall we exchange data. So there are different initiatives coming to exchanging data between hospitals. There is an initiative that we will make a European patient ID, which is UPID, which can be used for capturing this information. And the different part here is everything is compulsory. If it is a missing information, you need to provide the reason why it is missing. So we have HL7, which is the health language ontology or health language standard. Whenever there is a null flavor, you need to also provide this is missing, but because it wasn't provided, or this is missing because we did not ask for it. So you need to be in that detail that you need to provide the reason why the information is missing. And we capture this as a data catalog, which is called RDecor. Decor stands for data elements, code books, object identifiers, and rules. And yeah, so NICTIS is one of the Dutch health data exchange initiative from the government. And yeah, we capture this with NICTIS. And what it does is it really also facilitates many tools like ICRF generator, which is a Java program to basically generate case report forms in different formats like Castor, RedCap, OpenClinica, many other formats. And so again, we are just making the rules how to capture data. We are not making a goal that this is a database what we are forming. So a model is a mean, not a goal. You can, when you have these rules, you can pop out a database. If you like to have captured data as a database, if you like to capture as a semantic web graph, you can do that. So here is that using just a FAIRgenomes as publishing as a database. And these are like the cardinality relationships of that database. And yeah, so one of the other applications is MoleGenesis, which is a big database in Groningen. It's a city north of Netherlands, where we basically capture this. We now make the MoleGenesis database with FAIRgenomes rules, and then we can integrate with other research networks for like skin, carnioid. So there are different ERNs which capture data of a particular kind. We can combine it. In future, when we have, because we have these rules captured in data code books, I forgot to mention, it's XML based publishing of the rules. We would capture, we would make an ontology to basically find all the identifiers, missing identifiers. Currently, there's no identifier for basically capturing like karyotypic sex or NGS kit, and NGS kits are like, there's always a new kit coming up. So it is difficult to basically always keep an ontology which is on track. But the idea of ontology is that everything has an identifier. And then we'll have a demonstrator project for 1 million genomes, and then also making data in linked data platform. So we saw the success of FAIRgenomes in genomics, and we thought that it's very good that we reuse it for many other projects like the Netherlands Chrosomics initiative, where we want to, you know, advance beyond Chrosomics technology and develop FAIR data for every node of Chrosomics, that's genomics, proteomics, and metabolomics. and yeah, many different partners from Netherlands. And beyond the partners, the work we are doing in Chrosomics is also for basically data submission for, so whenever you now publish a paper, you either submit it to the, if it's genomics data, there's a requirement that you submit it to the European Genome Archive. If it is metabolomics, you submit it to Metabolite. If it is proteomics, you submit it to Pride. And the rules what we are making in Chrosomics, the idea is that we also facilitate a better data submission in these different initiatives. GH for GH really relies on phenopackets for capturing phenotypic data of a patient. And the idea is also that we want to make from phenotype to phenopackets, if it is in FAIR RDF format, we can transform this data to different formats. And one of the big initiatives for Chrosomics, what we have is that earlier our approach when this concept was starting, we always used to clean the data afterwards. Now we want to capture FAIR data at source once we have the rules. So the data should be FAIR from the machine it's generating, and then publish in a queue.
The real situation to the people who some way thinking that life is going on a different lines. So I think so if the FAIR data analysis would be there, people would be in a more real conception node that what is actually going in science, which is actually going in research, and actually being a part of health. Am I right, Gurnoor?
Yes. Yeah. Many times what in day-to-day life, like also for coronavirus, we had very big publications and then they were retracted. So what FAIR does is it really focuses on reproducibility of the data. And once we have that, we can always make this data available for, even for, if it is open data, we can make it available for people to actually analyze and visualize it as well.
Okay. So FAIR actually act as a filters and it makes the data more worthy for the people to be used.
Yes.
Okay. So Gurnoor, there is one question from student Utkarsha. She is asking that, so how the data which is available to you being cross-checked? So do you use certain algorithms? You use certain filters which make the data to be cross-checked?
So yes, one of the big challenges like of having FAIR, making data FAIR is like, you know, when you you start post-processing, you get the data and then later you kind of try to make it FAIR. So that is a process. It is a very difficult modeling process. And so I can give you an example for what we did in FAIRgenomes. It was like we combined all the stakeholders, different who were presenting their data and their metadata models. And then we were really focusing on like this data transformation. In regards to cross-checking, it's also like a post-process, but yeah, it's a challenging task. But it happens with the presence of a stakeholder if it is not FAIR from the source.
Okay. Gurnoor, now people are worrying about, okay, I'm sorry, please continue.
No worries. And when it's not FAIR, because what FAIR does is basically it's defining the rules of metadata, then you don't need to really cross-check. So if my weight and height, for example, is defined through some human phenotype ontology, then I really don't need to cross-check that it is weight or height, for example. But if it is not defined, then it becomes a cross-checking problem.
Okay. Now, Gurnoor, people are worrying about data theft. Hana? People are now worrying that the future time will be data might get lost, fine. Whether it's a health data, whether it's a research data, or whether it's a personal data. Do you people actually working in protecting that data so that people will feel little secure that if they utilize their data for any of the purpose?
Yes, ma'am. That's a very nice question and very meaningful question, I would say. So in regards to FAIR, the way we are trying to define this thing of like not having theft of data is with licensing information. So if it is in research, I can tell you there are like different type of licenses used. One is the Apache 2 license, which says, or MIT based license. These are licenses what are commonly used currently, which means that the data is open. But if there is something you're doing a post-processing analysis of using this data, if there's something nasty happening in your results, or you're doing a wrong prediction, I'm not responsible. That's basically the definition of Apache 2 or MIT based license. But there are licenses which restrict and limit people using the data, and it kind of tries to make a protection. In terms of infrastructure, when you have the data of a hospital is stored in a hospital, it is secure. It is like there is I would say a firewall of security behind it. And with like personal health train, what we do is basically we do the data communication with the help of APIs. So what APIs does is it just provides you, if you have an excess, you just are provided with the limited data what you ask the excess for.
Okay, Gurnoor,
okay. Now please, now let's
very last minute actually. We can disconnect in any moment actually. So I suggest whenever we are disconnecting, we will reconnect again to discuss few more things.
Yes, sir. It's like a break time for the kids. Gurnoor, are you there?
Yes, ma'am.
Okay, dear. So now I'm making the platform open for other candidates and participants and faculties to ask the questions, dear.
Let's hear a word from Sal, sir. Please, sir.
Yes.
Gurnoor, it is a very nice presentation. And do you find that your BTech BI course structure was fair enough on which you can build on, or any suggestions to make it more fair?
Be careful. Sir is using the word fair.
Yes, absolutely. I think absolutely, and absolutely, it was very fair. And I can give you a personal example. Like so, when I was studying like my bachelor's in third year, Tirstrad sir gave us a course called Algorithmic Bioinformatics, and the slides were exactly or very similar and complementary to the course structure I had in my master's. And actually I was like one of the good students for that course. And because like I had rarely studied these things before. In general, I think so. That's a very general thought I always have, is like nowadays the way we have these studying materials, and it is very standardized. And if the teachers are nice, if it is regular course conduct is happening at a regular pace, then you always benefit. And Jaypee did provide us with this nice platform that the teachers were very serious, and the course structures were very organized.
Okay, Gurnoor, there is a question from our student Janki. She's asking about that when we talk about fair genome data utilization, sometimes you use the information of patients which are having some concerns. So where the ethical issue comes when you use the patient's data?
Yeah, so my slides are not shared, but in regards to FAIRgenomes structure, like the modular data structure, we had two components. One was the general consent and one was the individual consent. So the general consent is a consent basically which defines that a government policy of like how the data should be ethically used, or communicated between different organizations. And then we also have individual consent, which defines whether the researcher is comfortable, whether the individual is comfortable with having this their data being shared or processed or being used for further research. So when we capture this information, all the communication of like FAIRgenomes data happens based on consent information.
Okay, I think so. It is pretty clear, Janki. I hope you now clearly understand that. It is not being misused because the person has to give the consent and the legal procedures are there, then only the data will be utilized. Fine. Okay, any more questions, please?
Okay, Gurnoor. First, I would like to compliment you for the beautiful presentation, and your slides were also very beautiful. Content was very good. And simply you described how simple data that we are normally doing, like first, second, third normalizations, or maybe fourth one also, but that is actually for the databases, but how it can be converted into a fair data with the principles of data mining and other aspects. So it was really good, and you simply explained everything. I hope the students learned few things from it and enjoyed it. My only concern is, like in Europe, normally these things are very well going on. So do we have some initiatives like this at other international platforms available?
Yes, sir. So one of the like, I can tell you like, so from, I'm not sure if it is a NATO initiative, but Global Alliance for Genomic Health, GA for GH, is a very big global initiative for capturing, and they also say that we want to capture data as fair. The European Bioinformatics Initiative, EBI. So EBI has a kind of slightly different policy, I would say, in terms of fair, that they, because they are very inclusive, and fair is also, at times, it's a bit tough to implement it because it is very well structured and defined. So the way they have is they want fair to be, you know, as a post-processing step, and we want fair to be as a source. But that's where we kind of differentiate. But we are in like talks with them to basically get these things fair at source and have the benefit of like capturing fair at source.
Okay, yeah, I have seen many omics types also that are also connected with fair data nowadays. So it's a very good implementation actually. I think now Dr. Jetta also want to ask you something. Dr. Jetta Shankar is here.
Surely, sir. It's mute, I guess.
Dr. Jetta, we cannot hear you. There is some problem with your audio.
Okay, Gurnoor, let me take that question till sir get joined. Gurnoor, people are always looking for Netherlands as one of the very nice, kind, and happy country. Now you come to the happy country of the world, Denmark, which is having the highest happy index. So would you actually interested in going to Denmark, or you would like to? Definitely opportunity must be very beautiful there. But what do you say that how Netherlands research patterns is different in case you have some experiences with your you mentioned about your previous institutes where you have experience of data analysis. Do you think so Denmark will be different from the Netherlands?
I think so, but they're very similar countries in general. But so I can tell you about the experience about Germany and Netherlands, how I had this. So I did my master's in Germany, and I think it was one of the best decisions of my life, because I also had admissions in like different colleges like John Hopkins and University of Melbourne. I applied all over the world, and the reason why I took Germany was there was also a senior called Sankalp Jain who was doing starting that time, and the reason why what I prioritized was that I'm not paying anything for my education. I'm
That's
getting
great.
exposed to the global world, and I'll be in a good city, in a good university. Maybe by the ranking, it's not like it was a very decent. It is still in like top 100 institutes, but not like the top 5 or these things. But yeah, I think if you have a good, so for me, like education in Jaypee was very complementary to education in Bonn. The difference comes in like projects or like the exposure. For example, in Germany, when you're studying there, you can also work in a company. So I was working throughout in 20 hours throughout my education in a company called a research company called Fraunhofer. And it was really nice in Germany, but it was like I think Germany really specializes on big initiatives and big infrastructures. And there are like in related to Bonn, like there are very big national infrastructures. And when I came to Netherlands, it was like Germany is, I would say, as big as Rajasthan, and Netherlands is as big as Himachal Pradesh. The nice part about moving to Netherlands was everybody knew everybody. After for networking and for collaborative research, I think so Netherlands was a very nice institute. And also like for PhD, I would say the Netherlands is a good, in Netherlands, PhD is considered as a full-time job. You pay taxes, you get good money. In Germany, you're still considered as a student. So that change I really liked, being a professional employee while doing a PhD.
So what is your suggestion to the... Sir, but a question. What is your suggestion to the students who are aspiring for taking admission overseas, Gurnoor?
Yeah, so I think in general, what I chose, and I really think that you should look for everything what is what concerns. It's not just the college, but the city you're going to, the courses you're going to be doing, the institute you're going to, and also how would be life there. For me, one of the prior reasons was that I don't want to be in a debt of like 60 lakh euros for 60 lakh dollars for 60 lakh rupees to basically start my life as a professional and then keep on paying that debt. So that was one of the reasons I took that time as a prior reason. But there are you should also look in scholarships and these things and apply everywhere and then put it on paper that what could be the best for you.
Good.
And
great.
In general, there are very similar countries.
Yeah, just want to tell you, ma'am, that I don't know, many students are fond of Netherlands. Priya is in Netherlands, Gurnoor is in Netherlands,
Yes,
and you know the MTech student Rajender, he is also doing PhD from
sir.
Netherlands. So 3 are currently in Netherlands.
That's great.
That is a good news, but hopefully you will not go.
No, sir. I will be here in JEIT. Don't worry, sir.
Yes, sir. That's great,
Dr. Jitendra, Jitu, you want to say something to Gurnoor?
When I joined, he's presenting well, sir. When I joined, he was just going to leave this place. But hello.
Yes, sir.
And secondly, sir को हमने नहीं जाने देंगे कहीं भी.
ये मेन बात है.
अच्छा.
But it's a nice presentation. That's all.
Thank you,
okay, thanks, Jitu. So I think that's all for today.
आप conclude कीजिए, sir. Today is your day, sir. Your student is there, so today please you conclude, sir.
He is a common student. He is for all of us actually. And thank you very much, Gurnoor, for joining us and sparing your time. And I am really sure that students and we all enjoyed this session. And be in touch, and we will take your services in future also.
Surely,
okay,
sir.
and whenever you are coming to India, I know you haven't came after your marriage. You went back from airport. I know it was a difficult situation, COVID and all that thing, but no problem. Whenever you are coming,
Surely,
and sir, I want to tell you that I joined his marriage also in Chandigarh. Me, Ragini and kids, we were there in his marriage. And it was a beautiful ceremony with everybody, and I met his batchmates and some juniors also from GUIT.
Yeah,
great.
So
Okay.
we enjoyed that moment, and whenever you are coming to India, please visit GUIT. We would like to have your interaction with us and students also. Thank you very much.
I would just say one thing nice about Bonn, University of Bonn. So we have like a generation of JUITians in Bonn, and that's why they were also in my
yes.
So I think from the second batch till I don't know, very down the line, we have like a lot of students.
Who is now there? Pranika was there, I think.
Pranika was there. So I was there. Swarit was there. Pranika was there. Mimansa was there. Isha Khandewal. She joined after I was there. So many students in, and also like Germany is a good, like gaining popularity in for masters right now. So
and they have good reputation towards our students also, I think.
A very nice reputation, sir. All have done very nice. Swarit Jasyal, he was my senior. He's now an associate professor in Japan. Pranika is doing a PhD in University of Zurich in Switzerland. Mimansa is doing a PhD in University of Bonn, but with Fraunhofer Institute. And Sankalp is now actually working in NIH. So he studied from Austria and now went to NIH. And Ashutosh Malhotra also did a PhD, and now he's in a very nice management position in a pharmaceutical company.
Okay, very nice.
That's great. So you're all well connected, Hannah?
Yeah.
Okay, so thank you very much.
So let's make it.
All the faculty members, Hemant ma'am, Gurnoor, and most importantly, the Synapse team. The Synapse team, kudos to that. They are doing wonderful.
Thank you, sir.
So thank you very much. Thank you, everybody.
Thank you. Thank you, Gurnoor.
Thank you, sir.
Take care, Gurnoor.
Thank you, Gurnoor. Okay.
Okay, sir.
Bye-bye.
Bye.
Bye, sir.
Bye.